morning, experts and scholars. Today, I would like to present our research, the co-occurrence of antibiotic resistance genes between dogs and their owners in families. Antimicrobial resistance, AMR, is a global public concern regarded by numerous well-known organizations, such as World Health Organization. Resistant bacteria already cause more than 700,000 deaths globally every year. It is projected to cause 10 million deaths per year at a cumulative global cost of £66 trillion by 2050. Increasing number and range of species of companion animals are kept in close interaction with human. Owners can gain significant improvements to health and well-being from the ownership of companion animals. However, companion animals have been described as potential reservoirs of antimicrobial resistance. From the One Health perspective, companion animals might be a source of transmission of ARGs, or antimicrobial resistant bacteria, to their owners. A possible transmission of MCR1, resistant to colistin, from companion animal to human had been reported. However, a comprehensive analysis of relationship of antimicrobial resistance between companion animals and their owners is lacking. Here, we firstly chose 13 owned dogs, 16 owners from 15 families as well as 22 kennel dogs and 22 Hadza to analyze the effect of close contact between owned dogs and owners on their gut microbiome, ARG and MGE compositions. We found that dog gut microbiota had higher abundance and diversity of antibiotic resistance genes, while owners had higher diversity of taxonomy. However, there was no difference in the total abundance and diversity of mobile genetic elements between dogs and owners. Meanwhile, we observed that samples of owned dogs and owners can be clustered into one group not only in their antibiotic resistance gene composition, but also in their MGE composition, while in their microbial community, they were separated. Owned dogs shared 70% of their antibiotic resistance genes, 82% of their mobile genetic elements with their owners. Furthermore, 81% of the species found in owned dog's gut were also detected in owner gut. We further compared the similarity of gut microbial community, antibiotic resistance gene and mobile genetic element compositions among the four groups using Jacket Index and found that there were higher similarities of the gut microbial community, antibiotic resistance gene and mobile genetic element compositions between owned dogs and owners than other pairs. Considering the close relationship of antibiotic-resistant bacteria detected in human and companion animals, we then picked 12 dog and owner pairs to compare their gut bacterial community, mobile antibiotic-resistant gene and mobile genetic element compositions. From the perspective of families, in terms of antibiotic-resistant gene types, the top 4 types in both dogs and owners were macrolide, tetracycline, aminoglycoside, and beta-lactam. The most abundant resistance types in both dogs and owners were macrolide, and its abundance was higher in dogs than in owners. The microbial community composition structured the resistant composition. At the family level, Bacterial was the most abundant in fecal samples, and its abundance was higher in owner than in dog. Dogs had higher abundance of Enterobacteriaceae and Peptostreptococci. Furthermore, we made taxonomic assignment of bacterial host of antibiotic resistance genes. At the family level, Enterobacteriaceae was the highest bacterial host and harbored most of diverse antibiotic resistance genes, including the majority of types in dog, while Bacteriaceae was the highest bacterial host in owner. Dogs had more Enterobacteriaceae than the owners, suggesting there might be elevated rates of gene transfer in the dog gut with higher abundance of those as they could promote the spread of antibiotic resistance genes. From the perspective of family, more than half of the detected antibiotic resistance genes were shared by dogs and owners. These shared antibiotic resistance genes had accounted for 88% and 96% in the detected antibiotic resistance genes of dogs and owners sample in the aspect of the total abundance, respectively. Meanwhile, we found that Escherichia coli was the shared strains between dogs and their owners by utilizing strain PHLM3. Variation partitioning analysis showed the shared bacterial community and moil genetic elements explained 85.27% of the total shared antibiotic resistance genes. The single shared bacterial community explained 40.43% of the variance, higher than that of shared moil genetic elements. We further analyzed the Pearson correlation of antibiotic resistance genes between dogs and owners, and found high correlation of macrolide resistance genes between dogs and their owners. The significant correlation was also seen in the subtypes. We also used binning method. After filtering low-quality metagenome assembled genomes, a total of 326 metagenome assembled genomes in owners and 215 metagenome assembled genomes in dogs were obtained. The phylogenetic analyses showed all metagenome assembled genomes belonged to 6 bacterial phyla. In the 541 recovered metagenome assembled genomes, a total of 58 metagenome assembled genomes were identified carrying mobile antibiotic resistant genes. The hosts of antibiotic resistant genes mostly belonged to the family Enterobacteriaceae in both dogs and owners. Finally, we got the following conclusions. Dog had higher level of antibiotic resistance genes, which may be caused by its higher abundance of Enterobacteriaceae. The close relationship between owned dogs and their owners had a profound effect on the modulation of their gut microbiota and promote the correlation of specific antibiotic resistance genes. That's all for our study. Thanks for your listening. I hope you will give us your comments and suggestions.